Welcome again to the Gastrointestinal Physiology lectures. Um, in the first part of these lectures, I talked about a definition of the gastrointestinal tract. I also talked about the control systems that orchestrate or regulate the various gastrointestinal functions: digestion, motility, absorption, and secretion. I talked about both the extrinsic and the intrinsic control systems, both of which contain nerves and endocrine secretions. That part, which consisted of 5 lectures, we called it General Gastrointestinal Physiology. Starting today, I will be talking about Special Gastrointestinal Physiology. As I explained it before in my introductory lecture, the first lecture, now I will be talking about the specific functions of the gastrointestinal tract. And the first function I would like to talk about gastrointestinal motility. This section will be divided into 2 parts. The first part, I will be talking about general concepts that will help us understand how the gut moves. And the second part, I will be talking about the specific motility patterns for each part of the gastrointestinal tract: the mouth, the pharynx, the esophagus, the stomach, small intestine, and large intestine. So, what are these general concepts that we need to keep in mind? First, generally, the contractile tissue of the gut consists of smooth muscle cells. This picture shows how the nerves supply such fibers. First, in A, which is to the left-hand side of the picture, we have one nerve that synapses with a fiber, muscle fiber, to cause contraction. I mentioned earlier that in the enteric neurons, there is a unique characteristic, and that is the presence of varicosities, which are swellings or bulgings along the axon of the enteric neuron that allows a neurotransmitter to be released for long distances on wider location, as shown in B on the right hand side of the picture. So the basic unit for contraction in the gut are smooth muscle cells, and please focus on the word most. Not all of the contractile tissue of the gut is smooth muscle cells. There are other places where you have striated muscle cells. There are places where you have striated muscle cells in the pharynx, in the oral third of the esophagus, and in the external anal sphincter. Now, we will continue on the special characteristics of the gut smooth muscle cells. First, we mentioned that they are supplied with enteric neurons that contain varicosities to release neurotransmitters on a wider area. What else is special about them? Well, the smooth muscle cells of the gastrointestinal tract are unitary type, which means that they are organized in units, and these units are connected electronically, meaning the electrical activity between them can move freely from areas that are called gap junctions, also known as nexuses. These nexuses allow for free movement of ions between the smooth muscle cells, which enable these muscles to contract at the same time. So the first characteristic of the smooth muscle cells is that they are unitary type, as this picture depicts. They are organized like units. And the second characteristic is that they are connected electronically with areas that are called nexuses or gap junctions, denoted here in this picture by the green and the red circle. On the bottom of the picture, there is an axon of enteric neuron that has vercosities where the neurotransmitter is released, and the smooth muscle cells are connected by gap junctions, also known as nexuses. These gap junctions make it easier for the ions to move between the smooth muscle cells along a wider area in the gut to make contraction happen on a wide range. Always keep in mind, please, that you have tens of meters of intestine. It is not just one organ, it is a pretty long organ which requires contractions at the same time in many parts of it. So it's very, very important. So two very important characteristics for the gut. First, the contractile tissue consists of smooth muscle cells, mostly, not completely. There are some areas that contain striated muscle cells. Another thing is that these smooth muscle cells are organized in units. Another characteristic is that they are supplied by enteric neurons, which contain veracosities. And finally, the smooth muscle cells are connected by gap junctions, also known as nexuses. Now, we need to understand how do they contract. We've talked about that before. There are two requirements for contraction to happen in the gut. What are these requirements? The requirements for contraction to take place in the gut is first, we have to have a slow wave coming from the interstitial cell of Cajal, or ICC. We have to have a slow wave that comes from the ICC. We're not only interested in this slow wave, we are interested in one phase from these slow waves, and we call that phase the plateau phase, which is number 3 in this diagram. So this is the first requirement to take place for the contraction to happen. The second requirement is an action potential. An action potential comes from the enteric neurons, either excitatory or inhibitory. Either excitatory or inhibitory. These are the action potentials that comes from the enteric neuron. Now the question is, Will there be contraction if there is an action potential coming from the enteric neuron? The simple answer is no. In addition to the action potential, we have to have a slow wave. Specifically, we have to have the plateau phase of the slow wave along with the action potential from the enteric nervous system or from the enteric neuron that in itself will cause the contraction. This is a recording from a smooth muscle in the gut that shows what we just talked about. First of all, on the top part, you have action potentials along with slow waves. Slow wave is present always in the gut. Action potential can happen when there is a stimulation. We can see in the bottom recording that contraction took place only when there were action potential, which is shown by the very small lines in the top picture, along with a slow wave. Slow wave plus an action potential will definitely produce a contraction, as the bottom recording shows. If there was only a slow wave by itself, like the one in the top picture, you can see in the bottom picture that there is no contraction at all. If there is action potential, like in the top picture, the very first recording on the very last left-hand side, you can see in the recording, again, there's no contraction. Contraction took place only when there were a slow wave plus an action potential, which is the two big recordings in the center of the upper picture that shows the contraction in the two central recordings in the bottom picture. So, contraction will happen when there is an action potential coming from the enteric neuron and a plateau phase coming from the slow wave of the interstitial cell of Cajal. These are very important requirements. So, now, if there is a contraction that took place by these stimuli, then what type of contraction can we see in the smooth muscles of the gut? There are also two types of contractions in the smooth muscles of the gut. One that is called phasic, and that lasts seconds, whereas there is another type of smooth muscle contraction, and it lasts for minutes or hours, and we call that tonic. So phasic and tonic. Phasic lasts for very short period of time. Tonic lasts for a longer period of time. Phasic takes place in the esophagus, small intestine, and in the gastric antrum, whereas the tonic contraction occurs in the lower esophagus, the cardia of the stomach, and the internal anal sphincter. This makes sense because these are all sphincters, and therefore they need to be tightly closed and for a very long period of time. You don't want the food to come in and out of the gastrointestinal tract freely, so you have to have a guard. You have to have a sphincter to guard it, to protect it, to keep it in. And that's why for these sphincters you need a tonic contraction to contract for several hours, whereas the phasic, you need it to contract for seconds. And this happens in the esophagus, small intestine, and in the gastric antrum. Okay, so now we've learned the requirements for the contraction. We have to have an action potential from the enteric neuron and the plateau phase from the slow wave of the ICC. We have two types of contractions. We have a phasic that lasts pretty short period of time, seconds, and a tonic, which lasts for hours. Now, we need to know what are the mechanisms for muscle contraction in the gut. Well, there also are two mechanisms of smooth muscle contraction. Electromechanical coupling, which basically changes in the membrane potential of the smooth muscle cells, and pharmacomechanical coupling or release of intracellular calcium. There are two mechanisms for muscle contractions in the gut. Those mechanisms are either by changing the membrane potential or by releasing intracellular calcium. One, we call it electromechanical coupling. The other, we call pharmacomechanical coupling. Again, change in the membrane potential or release of intracellular calcium. So when these mechanisms happen, what happens actually to the smooth muscle cell to make it contract? This what happens when there is a contraction in the smooth muscle cell of the gut. The muscle gets activated electrically, chemically, mechanically. Any stimuli, food can generate all of these stimuli, mechanical, chemical, and electrical. With that, you increase intracellular calcium. Calcium, in turn, binds with calmodulin. This phosphorylates myosin, and myosin interacts with actin, the two proteins of the smooth muscles. And when the myosin and actin interact, that's when contraction takes place. These steps must happen in a chronological order. They cannot, they should happen chronologically. They should happen one after the other. They cannot happen haphazardly. Otherwise, contraction will not take place. So, first you have to have a stimuli of the smooth muscle cell. Once you have stimulated the smooth muscle cell, you will increase intracellular calcium, which will bind with a protein in the cell in the smooth muscle cell. That's called calmodulin. Calmodulin, when it binds to calcium, it phosphorylates myosin, another protein in the smooth muscle cell. Once myosin is phosphorylated, it interacts with the other protein in the smooth muscle cell, which is actin. At that point, the interaction between myosin and actin creates the contraction. So first activation, then increase calcium in the cell, then binding with calmodulin, phosphorylation of myosin, and then myosin and actin interact. Once they interact, contraction takes place. So now, we talked about a number of things. First of all, we mentioned that there are special things in the gut that makes special motility patterns. We mentioned that the majority of the contractile tissue in the gut is smooth muscle cells, except in some few places it's striated, but for the most part it's smooth. These muscle cells, they're innervated by enteric neurons that contain vercosities, which releases neurotransmitters along the axon. The third thing that we talked about is that the smooth muscles themselves are organized in units. Not only this, but they are also connected electronically via gap junctions, also known as nexuses. Makes the ions move freely between these smooth muscle cells. Then we talked about the requirements for the contraction. What do we need to make a smooth muscle in the gut to contract? We need an action potential coming from an enteric neuron, and we need a slow wave, especially a plateau phase of a slow wave coming from the interstitial cell of Cajal. These are the two requirements. Now, when we have this, we have two types of contraction happening. We either have a phasic contraction, which happens for a pretty short period of time, or we have a tonic contraction, which happens for a long period of time. Now, when this happens, the mechanism by which the contraction takes place is through two mechanisms, either by changing the membrane potential, we call that electromechanical coupling, or by increasing calcium in the cell, and we call that pharmacomechanical coupling. So when these two things happen, not either or, it's both, they must happen, muscle contraction happen. It happens when the stimulation takes place, mechanical or electrical or chemical. Calcium is increased inside the cells. When calcium increased inside the cells, it binds to a protein called calmodulin. This combination will phosphorylate myosin. And at the end, myosin will interact with actin, the two proteins of the smooth muscle cells. When this interaction takes place, contraction occurs. There's one thing left for this story to be complete, and that is the direction of the muscle contraction. The direction of the muscle contraction can happen either in the ab-orad direction, meaning toward the anus, or in the orad direction, or orally, or toward the mouth. So you have either contraction that happens in the ab-orad direction, or you can have a contraction that happens in the orad direction. And you have the neurotransmitters which cause either the contraction or the relaxation to take place, because you have to remember that we have talked in the very first lecture in gastrointestinal physiology about the histology of the gastrointestinal tract, and we have mentioned specifically that there are two muscle layers in the wall of the gastrointestinal tract: an inner circular layer and an outer longitudinal layer. And we've talked about them with regards to moving the ingesta in the anal or in the ab-orad direction. We have seen this picture before. This is an illustration of how the peristaltic movement takes place. How does the gut contract in order for it to move the ingesta or the bolus of food in the ab-orad direction toward the anus? You have to have contraction of the inner circular muscle layer, which is shown here as thick muscle layer. At the same time and at the same spot, you have relaxation of the outer, thinner longitudinal muscle layer. We have seen this slide before. This action, contraction of the circular and relaxation of the longitudinal, will create two segments in the gut. These are the two segments of the gut as this picture shows. The first one in the bottom left-hand side of the picture, it's the propulsive segment. And the segment that receives the bolus of food is called receiving segment. So you have a propulsive segment and you have a receiving segment. in order for the propulsive segment to form, so to push the bolus of food in the aborad or in the anal direction, two things must happen: contraction of the circular muscle and relaxation of the longitudinal muscle layer. That's in the propulsive segment. Now, the receiving segment, they must have opposite action, so they can receive, they can accommodate the bolus of food that's coming from the propulsive segment. So what's going to happen is that the longitudinal muscle will contract, but this circular muscle will relax in order for it to accommodate the bolus of food coming from the propulsive segment. So again, we have propulsive segment and we have a receiving segment. The propulsive segment actions must be the opposite, the complete opposite of the receiving segment actions. The propulsive segment will have contraction of the circular and relaxation of the longitudinal, whereas the receiving segment will have contraction of the longitudinal and relaxation of the circular. And we explained that last time with the following slide. This is the slide where I mentioned that at any given point, which is denoted by the long vertical line in this picture, it separates the two segments from each other: propulsive segment from the receiving or the receptive segment. Propulsive versus receptive. If I have at that one point contraction of the smooth muscle, then in the receiving segment I must have relaxation of the circular muscle, and the opposite goes for the outer longitudinal. If I have relaxation at the propulsive, I must have contraction at the receptive. When you think about how these two muscle layers work, you should think about milking something from a tube, like the toothpaste, for example, or if you've seen the method by which people make sausage, this will also illustrate to you how the stomach or how the intestine moves, basically to move the ingesta in the anal direction. It's a milking process. And milking happens when one muscle contracts and the other receives, and the muscle contracts and the other receives, and so on and so forth, until you milk all the contents all the way to the anus. Now, so far, I have covered the general concepts and requirements for motility to take place. General concepts only. I did not talk about the specific motility patterns for each of the parts of the gastrointestinal tract, like the mouth, like the pharynx, like the esophagus, the stomach, small intestine, and large intestine. The next lecture, I will be talking about the specific motility patterns for each parts of the gastrointestinal tract. Again, this lecture talked about only the general requirements for gastrointestinal motility to take place. I have talked about the type of contractile tissue in the gut. That's mostly smooth muscle cells. There are striated in some areas, but mostly it's smooth muscles. I've talked about the special characteristics of the smooth muscle cells. They are unitary. They are organized in units. They are supplied by enteric neurons that contain vercosities to release neurotransmitters on wider areas. I've also talked about how smooth muscle cells are connected electrically by the presence of nexuses or gap junctions, which allows the ions to move freely between these smooth muscle cells. I also talked about the requirements for contraction. We have to have two important elements. We have to have an action potential from an enteric neuron, and we have to have a slow wave coming from the interstitial cell of Cajal, especially a plateau phase. When we do that, we will have two types of contraction. We will have a phasic contraction that will last few seconds, and we will have a tonic contraction, which will last even hours, like in the sphincters. Then we've talked about the two mechanisms by which the smooth muscle cells of the gut contracts. We said that two things must happen. One is change in the membrane potential, action potential of the membrane, and that mechanism is called electromechanical coupling. And then when this happens, calcium is released from the cell, and we call that pharmacomechanical coupling. When this happens, this calcium binds to calmodulin, which phosphorylates myosin, and myosin then interacts with actin, the muscle contracts. Finally, we've talked about when this contraction takes place, the process is called milking of the ingesta, milking it all the way from the mouth to the anus. And in order for you to milk that, to squeeze, if you will, the ingesta all the way from the mouth to the anus, at every point of the gastrointestinal tract, you create two segments, a propulsive segment and a receptive segment. A propulsive segment and a receptive segment. At every point, you have to create these two segments in order for you to milk the ingesta in the anal direction. And when you do that, you contract the circular muscle and relax the longitudinal muscle in the propulsive segment, and the opposite action happen in the receptive segment. you have relaxation of the circular muscle and contraction of the longitudinal muscle. That's how the receptive segment will accommodate the product moved by the propulsive segment, which is food in this case. This is a summary of what we've talked about today. Next lecture, I will be talking about the very specific motility patterns for each part of the gastrointestinal tract.